My name is Moise, and I am a graduate student in Bevan Engelward's lab at MIT, and I study probiotics. Probiotics are becoming increasingly common, both in how many are offered and how much they're being recommended for different uses. Just a few years ago, there were only a few options for different probiotics you could take, but now you could walk into any pharmacy or grocery store and there are shelves full of them. They come in all sorts of types that can have different formulations and benefits. For my studies, I use a variety of methods to study whether probiotics can change the potentially harmful effects of exposure to different kinds of chemicals. One chemical in particular that I study is N-nitrosodimethylamine, or NDMA. NDMA may cause cancer in humans, but it is a very potent carcinogen in rodent models, especially in the liver. Probiotics have been shown in previous animal studies to suppress the development of cancer due to other risk factors, such as genetics or even lifestyle. I wanted to see if it could have an impact on the formation of cancer due to NDMA exposure. For my research, I am interested in something called the two-stage model of cancer development. This model describes how normal, healthy cells could eventually lead to cancer. The first stage is initiation, where a cell gains a mutation that may provide some sort of advantage. These advantages might include dying when it shouldn't or not dying when it should. The second stage is promotion, where the cell has that advantage, it gets chances to grow and divide and gather more mutations that eventually change its behavior. While NDMA is a DNA damaging agent that causes mutations which can contribute to initiation, it also induces inflammation. Because probiotics have been shown to have some anti-inflammatory properties, I was interested in the impact of probiotics on chemically induced inflammation. After inducing inflammation, I looked at how the liver responded with or without probiotics treatment. In particular, I examined how probiotics change the patterns of cell division throughout the tissue. Not only did probiotics change when cells grow and divide, they also change the pattern of division across the liver, especially in male mice. I also saw differences in the amount and types of immune cells that are recruited as part of the inflammatory response. I also looked at the pathways that are activated or suppressed by liver inflammation. Probiotics appeared to initially enhance immune cell activation and recruitment, but quickly suppress it again. Inflammation activated pathways associated with DNA damage, especially with double-strand break repair. Probiotics actually dampened activation of these pathways. Overall, probiotics don't fully prevent the damaging effects of NDMA exposure. However, they do alter the short-term inflammatory response and the way that tissues respond to chemically induced damage. Changes in the immediate response to damage could have an impact on the long-term outcome from an exposure. These studies also contribute to the growing body of work on the potential beneficial impacts of probiotics on health and disease.